So Paul and Caitlin, if y'all want to come up here, that'd be great. All right, friends online, we're going to get started in just a minute. This event is being interpreted into Spanish online and closed captioned. So if the people here in person, if you want Spanish interpretation, you can go outside and join on Zoom, I suppose. The people who are on Zoom watching, you can change your channel over to the Spanish language interpretation if you'd like. It's also being closed captioned into English and Spanish on Zoom, so that those accessible resources are available for you there. And if you're watching on Facebook, it's not too late to join on Zoom, so you can get all these resources. But hello, my name is Caroline Nickerson. I'm going to be moderating today's event. I'm representing SciStarter. I'm really grateful to the network of the National Library of Medicine for making this event possible, as well as to the All of Us research program. What better way to celebrate National DNA Day than coming together for a fun Zoom event and in-person event where we can talk about the latest and greatest in genomics research and genetics research. And you can learn all about the All of Us research program, as well as some other citizen science activities you can do to turn your curiosity about the world into real scientific impact. So anyone can do it. Anybody of any age can get involved with citizen science. And the All of Us research program is a great opportunity for people living in the States who want to contribute to the future of health research and also want more information about their own health. We have a panel lined up for you today where we're going to talk all about different topics that fall under the umbrella of DNA, genetics, genomics. I'm going to go ahead and stop sharing for now, just so the people online can see our panel. So I'll let them introduce themselves before I get to an intro about Citizen Science Month, which runs all through April and is part of today's event. But if you want to go first.
Sure. So I'm Sandy Borowsky. I'm a physician and professor at UC Davis. We're one of the sites recruiting for the All of Us study, and I serve as the principal investigator here at Davis.
Awesome. And then if you want to pass the mic to Hugo. All right.
Hi, everyone. My name is Hugo Campos, and I am a participant in the All of Us. I'm a patient, I'm a person living with a genetic heart condition. You'll hear a little bit about it in a little bit.
Fantastic.
Thank you for coming.
I'm Paul Lott. I'm an assistant project scientist at UC Davis here and a proud participant of the All of Us study. So we're excited to tell you a little bit about the research we're doing and using All of Us.
Woo.
Hi, my name is Caitlin. I'm an undergrad at UC Davis, and I'm going to be presenting with Paul today a little bit about our research.
Yay, we're delighted to have you. And for our friends on Zoom and watching on the Facebook live stream, we're paying attention to y'all too. Don't be shy about asking questions about anything you're curious about. If you have a thought to share, put that in the chat. We really do want to hear from you, and we view you as full participants in this event as well. And for our friends in person, remember, the people online can't hear us if we don't talk into the mics. So I'm going to get my slides shared up again for a brief intro. So we'll just go share. All right. And if you want today's slide deck, and this goes for the people online, in person as well, if you want any of the links from today, if you want to look over something and learn a little bit more about it, or you just want to take a closer look, you can go to this link, scistarter.org/go/National DNA Day with the dashes, and you can download today's slide deck, get all the links from today to fun videos, to handouts. We have, some talks geared up for you, but we also have bingo that we're going to play in relation to All of Us. Everybody loves bingo. We have another handout for you. And of course, the citizen science activity, stall catchers, you can do any day of the week, anywhere in the world if you have internet access, and classify blood vessels for Alzheimer's researchers to help move the search for a cure forward. It's also a really fun project. You can get points, you can gamify it, you can compete against your friends and family to make the world a better place with research. So all of those links are going to be available to you in today's slide deck. So we'll go to the next slide. So I'm Caroline. I'm an advisor at SciStarter, as I mentioned, but first and foremost, I'm a citizen science enthusiast. So we'll go to the next slide. So what is citizen science? Well, it goes by a few different names. Some people call it community science. Some people call it neighborhood science. I've also heard public participation in scientific research. That is a mouthful. But basically what it is it's real science that you can do. It's collecting or analyzing data for researchers to answer questions that we all want answered, to discover things that we all want to know. It can be water quality research. It can be astronomy research. I've worked with citizen scientists who have discovered spiral galaxies. You can do it outside, document different plants and animals. You can do it from your smartphone by playing an online game to help Alzheimer's researchers and annotate the data for them. Citizen science really gets at data, which is all about information, right? It's, you know, words and pictures, it's numerical measurements like temperature or inches of rainfall. It's citizen science is about collecting and analyzing this data. And by analyzing, it means we just take a closer look so we can learn something new about the world. Citizen science is about creating new knowledge. So if you think you don't like science, if you're a curious person, you probably do like science, and you can do this science at any age with citizen science. So we'll go to the next slide. So at SciStarter, one thing we always like to say is you can turn your curiosity into real scientific impact. So SciStarter is where you can go to find thousands of different citizen science opportunities, where you can, like I mentioned, monitor biodiversity, identify different plants or animals, where you can help with health research. Researchers have added these projects to SciStarter for you to discover, and you can go to SciStarter and make an account to keep track of all the different projects you're joining. And it's really fun. You can, you know, it's bragging rights, right? You can look back and say, wow, I contributed to 40 different citizen science projects this year, and I'm a good person. I've done really good things, but it's also fun too. You get to learn new things about the world and really investigate your own community. And you never know what you're going to find. We'll go to the next slide. So every April, we do Citizen Science Month. We're in the home stretch. I love that I get to do this event to really ring it all in. It's just an excuse to celebrate. You know, you can do citizen science any day of the year, but with Citizen Science Month, we kick it up a notch. We do a bunch of events. There are still webinars and online events you can join if you go to citizensciencemonth.org. But there are social media graphics you can find where you can share your excitement. There are so many cities, and at citizensciencemonth.org, you can find it all. And it's made possible with the support of the network of the National Library of Medicine, the All of Us Research Program, Arizona State University, SciStarter, and many other partners from around the world. So go to citizensciencemonth.org to check that out. We'll go to the next slide. So those are all the resources that you can find online. This event was advertised there, but you can also find the projects we're talking about today. You can find stall catchers, that Alzheimer's research project we're going to do, and many more opportunities as well. Next slide. That's the event calendar I mentioned. Today's event is up there. But yeah, people from all around the world were really getting excited this month. It was cool to see. We'll go to the next slide. And there's the featured projects page. So scistarter.org/NLM, that stands for National Library of Medicine. Stall catchers is featured front and center on that page. You can find it. You can also find the All of Us link, but if you want to go directly to All of Us, you can go to joinallofus.org. But this is where you'll find the featured stall catchers project that we're talking about at the end of the event. Next slide. So April is Citizen Science Month. We're delighted you're here, and we're delighted to be in conversation with you today. But first, we got some exciting talks. So we'll go to the next slide. So we're going to pass the mic to Sandy, where he's going to talk a little bit about the All of Us research program.
Thanks so much. So I'll start with a few questions to make sure everybody's awake. How many of you are participants in the All of Us study? And if you're online, go ahead and raise your hand.
The online people, there are a lot of people in the room who participate in All of Us. Join their ranks. If you're watching and you're inspired, join them.
So my first message is thank you. Thank you for joining the All of Us study. You probably learned a lot about it along the way, but I want to give a brief introduction from my perspective and let you know why I got involved in the study and why I think it's so important. So first of all, if you go back and look at how we practice medicine and what is the data or evidence that we use to make recommendations for how to treat high blood pressure or how to treat asthma or any number of diseases, we have limited data to do that from. So essentially, all of the recommendations that we use for high blood pressure and heart disease prevention all come from a very famous study called the Framingham study. Who's heard of the Framingham study? I have a couple of people.
Oh, we're having mic issues, so we'll switch over to you. Okay, here you go.
It went yellow on me.
Oh, no worries.
It went yellow on me. That's still yellow.
I think.
Is yellow good? Or do I need green? I can talk right into that thing.
Maybe ask if they can hear you online, because if you
The
press
lights
the
on
button.
this one off too. Can you hear me online?
Oh, they can hear you. We're good.
Okay. So the Framingham study was 4,000 people in Framingham, Massachusetts. And Framingham, Massachusetts is sort of a upper middle class suburb of Boston. And as you might imagine, those 4,000 people are not necessarily representative of the whole US population. And so all of our recommendations come from that population. And yet, the more we learn about medicine and the more we learn about better ways to treat people with different diseases or different susceptibilities to diseases, the more we know that one size does not fit all. Different recommendations apply to different people, to different populations of people, and it depends really on who you are. And so the big goal of this All of Us study is to figure out who you are. So what makes you? So a big part of what makes you, we're going to talk about today, is your DNA. You, of course, inherited your DNA from your mother and your father and their mothers and their fathers and all the way back through your ancestry. But that's only one part of who you are. You are also who you are because of where you live, who your friends are, what your job is, how stressed out it makes you, what you eat, what your activities are. So all of these are important things in figuring out who you are and therefore what's going to be the best recommendation for you to hopefully prevent disease. I'm big into prevention, but even when you get disease, treat that disease. So that's really the goal here. So when this project was conceived, they decided that they wanted to represent all the diversity of the United States. Doesn't matter who you are. So based on that, anybody in the United States can join this study. You have to be 18 or older for the moment. So if you're 17, call us up when you turn 18. We'll sign you up. And you can't be currently in jail. So if those of you who are online are currently incarcerated, you have to wait till you get out and then you can join the study. But those are the only limitations to joining the study. Otherwise, everybody's eligible. You do not have to be a documented citizen. You just have to live here in the United States. And that to me is super valuable as a way of saying, we care about everybody, not just the 4,000 people in Framingham, but everybody in this country. There's one other really big thing that is an underlying philosophy of this study that we, especially Hugo and I, really strive to live up to. And I'm not sure we always do a great job, but we do believe that when you join the study, you are not just a participant in the study, you are our partners in the study. And we can learn as much from you as you can learn from us. And that's a big part of today too. We really want to encourage you to be an active participant, to learn from your own DNA, but also to ask questions about the whole population. And so we have, with all the appropriate sort of regulatory protections so that your data is secure to you, we've still been able to open this researcher workbench to open up the ability, without knowing who individuals are, but to know what the population looks like. How many people carry this gene change or that gene change or this combination? It can be very complex combinations. And you're going to hear a little bit more about that today from Paul and his group. So I hope you enjoy today. I want to encourage you to be an active participant and partner today and ask questions. So I'm happy to field any questions, but I don't want to spend too long talking because these guys are going to give you some really good examples to get you thinking.
Yay, awesome, super like that. So we did get one, thank you so much, that was awesome. We did get one question to start. I feel like it's a good time to ask. So someone asked, how does a single study based on 4,000 people determine medical treatment recommendations? If you want to take that.
So what we learned in the Framingham study was important. We learned things like, if we reduce your blood pressure with medicines, and we have very good medicines to do that, we also reduce your chance of having heart disease, having a heart attack, or having a cerebral vascular, a stroke that can relate from high blood pressure. So we learned that with evidence that there is a connection between these different disease states that leads to further disease. And that was a really important and eye-opening kind of thing for us. But we didn't learn how to treat hypertension, for example, in African-American men, where it's much different and actually much more difficult to treat hypertension in African-American men as compared to what we learned from the Framingham study. So I shouldn't sell it short. There have been lots of studies since then, that have incorporated specific populations to ask those questions so that we can learn sort of one at a time. But as you guys know, we are now in this, what some people call big data era. And so now when you go on Google or Facebook or Instagram, they're collecting data on you because they want to know what ads to show you. Well, we're kind of doing the same thing, but we're collecting data on you so that we learn how to treat you better and how to keep you healthier. And so we also are a lot more careful about keeping that data secure than they are in the commercial world, which is nice. But you know, this big data collection and being able to sort through lots and lots of people, but also lots and lots of data elements on each person is something that we need to take advantage of to improve people's health. And that's what we're trying to do.
Fantastic. And before that was such a great start to the event, and happy National DNA Day, everybody. We're happy to celebrate with you, and that's a great way to celebrate. To also start on a celebratory note, we had some exciting news to share. Hugo actually flagged this for me earlier, but with all of us, they've made nearly 250,000 whole genome sequences available for scientists to study. So that's the biggest and most varied genomic data set of its kind in the world. So huge celebratory note this Citizen Science Month. Yeah, we have a question in the audience really quick.
So I don't know if this.
Yeah, that way the people on Zoom can hear us too. We want our Zoom friends to feel part of the event.
Yes, this actually ties in with what Dr. Borowsky was talking about. So 250,000 whole genomes. What are the demographics? Because I know there are other studies like or biobanks where you might have 500,000 plus participants, but they're all of European descent. So I'm curious what the breakdown is.
Yeah. So does anybody want to take that one to start? All right, let's go for it.
I gave Paul a slide on that.
Okay, I'll wait. I also just gave a talk on something very similar.
But I will tell you, we have worked very hard to ensure that all the groups that are underrepresented in biological research are well represented here. And there are, some challenges with that for sure. The Native American populations, this is a big deal because there's a lot of trust issues between the tribes and the NIH, and well-deserved. But we're trying to work through a lot of those issues. But you'll see a slide of what the demographics look like in a little bit.
Yeah. Yes. Spoiler alert, everybody. Yeah, well, we're gonna get to that. I think right now, Paul and Caitlin are up next. Just a reminder to people who are here in person, we have snacks for you. So if you get hungry while Paul and Caitlin are presenting, you can grab some snacks. People watching on Zoom, unfortunately, we do not have snacks for you. And also a big thank you to the Sacramento Public Library for providing this space for people watching on Zoom. It's like you're here with us. But yeah, up next, we got Paul and Caitlin talking. So I'm gonna share the screen again so the people on Zoom can see it. So we'll share. Go. So Paul and Caitlin, whenever you want your next slide, just say next slide. And here's the mic.
Thanks. Yeah, so stand up here. Okay. Yeah. Pass the mic. So yeah, I just wanted to say we're excited to really be here and to be able to tell you about our research and how all of us is really helping us to accelerate our research and make findings that address cancer and health disparities, improve cancer for everybody in our communities. So next slide. So just a quick introduction. I know we introduced ourselves a little bit before, but so I'm Paul Lott. I'm an assistant project scientist at UC Davis Genome Center. I've been studying human genetics for the past 20 years. Had been in Luis Carvajal-Carmona's lab since 2013. As a scientist, I was a trained geneticist, but I worked mostly on the computer. So I'm often known as a bioinformatician working with large data sets. So.
Hi everyone, my name is Caitlin. I'm an undergraduate at UC Davis. And I joined the lab in the fall of my junior year, and I'm currently in the last quarter of my senior year. And today we're really excited to share a little bit about our research and how we're using all of us to make these discoveries. So I'm going to start by giving everyone a little bit of background on genetics research. So in 1953, in a lab in Cambridge, England, Rosalind Franklin, Francis Crick, and James Watson discovered the double helix structure of the DNA. Oh, sorry.
No worries.
And DNA is the molecule that's contained in all of our cells in order to provide instructions to our bodies on how to develop, survive, and reproduce. In the past 70 years, we've learned a lot about the processes that use DNA to perform various functions. And one example of this is creating proteins, which are important for things like muscle contraction and creating antibodies and many other things. Today is also the 20th anniversary of the completion of the Human Genome Project. And this was the first time that scientists were able to create a whole reference DNA sequence for all of our chromosomes. And having a reference genome meant that scientists could start to understand why differences between individuals exist. One example of these differences is explaining why maybe for some individuals a protein is made differently or not made at all. Here, there are two DNA sequences, and you can see that if a C is substituted for an A base pair, this causes the protein to be truncated. So everyone's DNA differs a little bit from the reference, and this is normal. Some of these variations you inherit from your parents, and others just happen by chance throughout your lifetime. While most of these changes are harmless, some variations can lead to more significant changes, such as the one that's shown in this slide, and this can lead to various disorders. This variation is one of the tools that geneticists use in order to understand disease. Next slide. Sorry.
No worries, I understand.
And as geneticists, we're primarily interested in how genes influence and impact our health. However, things such as the environment and the choices we make also impact our health. And this may be things that other scientists will look into. We're excited to be with everyone here today and celebrate National DNA Day with all of us and share some of our research in order to help you all understand why as researchers, Paul and I are participating in the All of Us collaboration within our community.
Yeah, so just to kind of start off, tell you a little bit more about background about the lab that we work in. So Luis Carvajal-Carmona, here, shown here, established his lab at UC Davis in 2012. And so really the research focus of his lab is cancer genetics. So what's the causes of cancer, how do we treat cancer, and focusing on cancer in Hispanic and Latino populations. And so we're really interested in understanding many different types of cancer. You can see here breast cancer, colorectal, stomach, thyroid, ovarian, so a very wide variety. We're also, very involved and in developing the cancer health disparities in Hispanic and Latino populations, and addressing those because there's a huge disparity, and I'm going to talk a little bit more about that in just a second. As well as the lab is also involved in building the cancer health equity community in the Central Valley here in California, helping people to have outreach to learn about cancer, the risks of cancer, and different things. So our research is very collaborative. It's really international. We do have other labs that we collaborate with here in the US, but also Mexico, Colombia, Chile, Uruguay, Argentina, Portugal, Spain, and the United Kingdom. So it really is just an international collaboration. Next slide. So when I talk about health disparities or research disparities, we kind of hit on this a little bit before. I just want to take a second kind of to describe that more in depth, and so that you can understand what we mean. So health disparities arise. So essentially the adverse outcomes of some result, right? And in this instance, health or the research that leads to adverse outcomes of our health. And those essentially have a background in the racism, discrimination, and segregation that we have a history of in the world, in our country. And those lead to essentially structural inequalities and social injustice, right? That affect us all, and affect some more than others. And essentially they affect, a lot of these social determinants of our health, right? So our socioeconomics, which is our education, our income, our employment, our clinical, which is essentially what access do we have to healthcare, right? Our environmental, where we live, what we breathe, the air we breathe, affects our health. The cultural, our health beliefs, our health-related attitudes. And then we're going to talk a little bit more today, which we're a lot is focused on this, the biological, right? Genetics, epigenetics. We also have, behavioral, right? So diet, tobacco, exercise, things like that affect our health. And the psychosocial, which is stress, mental health, those things. And so essentially, as we think about how those all act together, as then Dr. Borowsky mentioned before, right, the Framingham study, right? The 4,000 people led to great discoveries, but a lot of people weren't represented. And so those development of those discoveries and detection and treatment and survivorship are really, helping disproportionately, a different segment of it, not the whole population. And so there's a lack of diversity in cancer research and care in the workforce, and that's what it leads to, and ultimately adverse outcomes. Next slide. So kind of in a research aspect, this is kind of the diversity that we see right now in one of the big disease study databases in the world. So the GWAS catalog, that's a catalog that I use a lot. But what you see here is, right, European people of European descent or origin ancestry are represented as 95% of that data set. Asian populations only make up 3.3, and the rest of the categories here, African, African-American, Afro-Caribbean, Hispanic, and others or mixed races, make up less than 1% each in that database. And this isn't representative of our community, our world. And so we see there's a huge research disparity, and without that representation of the people in our community, we're not going to be able to address the health issues and the risks and the disparities of those individuals. So that's one of the reasons we're really focused on addressing health disparities in our lab. Next slide.
I'm going to be talking a little bit about gastric cancer. It's the fourth most common cancer worldwide. Each year, there are 1 million individuals diagnosed with it, and around 770,000 deaths per year. Gastric cancer cases tend to be broken into two major categories. So 80% of individuals with gastric cancer have no genetic component, whereas 20% of cases have a genetic component, which means cancer runs within their family. Within this 20%, there are 3% of individuals that have mutations that basically lead to a very high likelihood of them developing gastric cancer in their lifetime. So at its earliest stages, gastric cancer starts as a tumor in just the inner lining of the stomach. At this stage, it's much easier to treat because doctors can just remove the part of the stomach that's affected. However, when you get to the later stages, the tumor can go through all the layers of the stomach and also spread to other parts of the body. And by this point, there are a lot less treatment options available, and treatment outcomes tend to be worse. Gastric cancer also, in particular, tends to have a pretty poor prognosis because symptoms don't tend to show up until you get to the later stages. In particular, Hispanic populations also tend to be diagnosed with gastric cancer at later stages, so their outcomes tend to be worse. Next slide. And within gastric cancer itself, there is a clear health disparity. Here on the graphic on the left, you can see that stomach cancer incidence is roughly 2 times higher, and death rates are more than 2-fold higher in Black individuals rather than white individuals. And then on the graph on the the right, you can see that stomach cancer rates for 100,000 individuals are twice as high for minority populations. In particular, our lab is focused on addressing this disparity within this area.
Next slide. So I'm going to tell you a little bit about our current research study that we are currently working on and how we're using all of us. We previously, so in 2013, when I joined the lab, we had about 32 samples of gastric cancer in Hispanic patients. And we looked at the DNA of those and identified some variants, some mutations in the DNA or differences in the DNA that we thought may cause an increased risk. And we were able to look at other data sets through contacting other researchers to see, hey, can you look in your data set for this? Can you look at this? And this took about 4 years in order to actually come. And what we found is that indeed, mutations in these DNA repair genes, BRCA1, LB2, and RAD51C, increased the risk of gastric cancer, and it was in these families. And so, building off that study, in 2017, we said, hey, there's something here. Well, how prevalent or how frequent are these mutations? How often are DNA repair genes involved in these in gastric cancer? Our paper that we published was one of the first that was published on this. It had previously not been known. And so we looked and said, okay, well, let's spend the next essentially 5 years gathering subjects, cases, individuals that had gastric cancer before the age of 60. And we sequenced 513 of those individuals. And what we found so far in that study is that over 1 in 8 of those individuals carry an inherited variant in the DNA repair gene. So we see that, and this is known known variants that have been known to be pathogenic, right? The thing is we've got thousands of variants in our data set that we just have never seen before. They are so rare, and the data sets aren't available that looking at Hispanic populations. So we don't know how prevalent these are within the populations. And so we just have to look in other data sets. So another things that we've identified is that really the types of cancer that we're seeing in these individuals is the more aggressive forms. It's what we call genomically stable diffuse. We're seeing somewhere in the ranges of 60% of our individuals in our study having this type, which is significantly different than what other studies in European data sets. The Cancer Genome Atlas did a study in the gastric cancer, and it was almost exactly it was much less frequent, about a third less frequent in their population. And so we're seeing this, and so our goal really now is that, okay, how do we identify these risk factors? How can we move from the few that we know for 1 in 8, and we have 1,000 that we don't know, and so we really want to be able to do that so that we know if they're associated with gastric cancer, so that we can help develop better screenings, better treatments for these individuals, so that they have better outcomes. Next slide. So to kind of help you understand how all of us is helping research and kind of the huge paradigm shift that this is for research. Why, as a researcher, I am so excited about all of us and how it is going to transform research for me and Caitlin and future researchers, all the whole research community. I have kind of a little activity, and for those on Zoom, it will be apparent you won't have your honey pot, but next slide. But you can participate with us and kind of look at the slides and understand. So I want to kind of start off by saying, I'm going to use kind of a little metaphor. When I like to think about this, I like to think of Winnie the Pooh. A favorite story of mine is growing up as a little child was The Adventures of Winnie the Pooh. And we all know that Winnie the Pooh, he loved to eat, and not just any food, he loved honey. And so most of his adventures were about him collecting honey for his honey pot and then eating it, right? And it was very protective of his site. And so I'm going to kind of use the honeypot metaphor, kind of relating to the research paradigm of the past and help you to understand how all of us is changing that paradigm. All right, next slide. So as a researcher, we're interested in, just like Winnie the Pooh is interested in honey, I'm interested as a geneticist in variants and human genetics, understanding how our DNA leads to, who we are, what leads to disease, what increases our risk of disease, and how we can actually use that. So when I go out, right, I'm excited. I want to put things in my honeypot, but as a single researcher, I'm very limited, right? I'm limited to those that are around me. And so when I go out, I find, for example, for this study in stomach cancer, I find individuals, hey, they're younger, they have gastric cancer, it's Hispanic origin. I'm going to put them, I'm going to add that to my honeypot, essentially my data set, right? And so, next slide. So I've handed out in the audience here just a couple little honeypots to people, and they have different shapes, right? And so each of you, as even the ones on Zoom, you can think of, okay, what would be your honeypot, right? What disease? Would it be Alzheimer's? Would it be high blood pressure? Would it be heart disease? Mental health, right? Maybe depression or bipolar disorder or something, right? So, and just like you could be, you're a researcher, right? With all of us, you're a researcher. You can fill that honeypot. And so essentially, as a researcher, I like to fill my honeypot. Essentially, you can see the different shapes and the different colors. The different shapes and the different colors just kind of are the different attributes of those things that I'm interested in, right? So for me, gastric cancer, mutations in the DNA. And so, next slide. So everybody here is a researcher. So what are some of the diseases that you might be interested in? All right.
Get as close to that mic as you can so our friends can hear you.
Narcolepsy.
All right, yeah. That's great.
Hemophilia.
Hemophilia.
Breast cancer.
That's great. Alzheimer's, great. So yeah, so we're all interested in different diseases. We have our honeypots and we filled them up, right? All right, next slide. So I want to ask a question now. So basically, looking at your honeypots, you have a gray triangle on your honeypot. How many gray triangles do you think are in this room?
2.
2. Can you tell?
No.
No, because you're just looking at it. All right. What might you have to do to find out if there's more honeypots or with a gray triangle?
Get a larger sample size.
Yeah, get a larger sample size. All right. So let's actually, can you pass your honeypots to the center? Let's get a larger sample size, right? Let's combine our studies. All right, so you want to come up just real quick? Thanks. Let's just lay these out and see how much easier it is, right? How many gray triangles do we see now?
4.
4. Well, that was much easier, right? So it's so much easier when we collaborate as a community and bring our data sets together. Now, one of the things you'll notice with these is, did anybody see their honeypots change? No, they're a piece of paper, they're a printed piece of paper. Right? That's a problem with our data sets. Oftentimes, as a researcher, I'm interested in a couple attributes, and I go out and collect those. And I collect those samples. Well, I can't then pass that off to, say, Dr. Borowsky, and he's interested in something totally different, or somebody else is in the narcolepsy, right? my cancer data set may not be applicable to them. I didn't collect the right data. I didn't have the right attributes. Plus, today I might not have skin cancer or stomach cancer, but the next time I go into the clinic, I might. So if it's changing, it's very dynamic, right? These are not dynamic. These aren't updating. These are very static. And so that kind of illustrates kind of an issue with the past research paradigms. Often our data sets were so limited, small. There are honeypots, but really in order to be able to make these big discoveries, we needed to combine them together. So next slide. So Caitlin's going to tell you how all of us is really changing that.
This is where all of us is addressing the old research paradigm, rather than each researcher having their own limited honeypots of data. All of us is empowering us as a community of participants and researchers to build rich community data sets. And these data sets better reflect our community's diversity that allows us to ask important questions and address health disparities to accelerate the discovery of better treatments. Comparing the all of us data set from this month versus the GWAS data set that Paul talked about earlier, we can clearly see that the all of us data set contains is much more representative of our population and contains more data on minorities.
So this month, we received an email about the research workbench, and it was mentioned here earlier that 250,000 genomes. Is that huge? That's really, really huge. And so I wanted to talk to you a little bit about kind of the data, right? So this data is community, right? It's available to all researchers right now that we can log in and start to mine this. It's very data rich. Instead of just a single shape or color, we have survey responses, over 400,000 survey responses, physical measurements, 300,000 genotyping information, electronic health records that are updated when we go to the doctor. Maybe, and I get a diagnosis for skin cancer. Well, that's updated. That goes into this data set. So it's dynamic. It's very, very rich. And so that a researcher can ask these questions that before, as a researcher, I've been frustrated when I want to ask this question, but I don't have the data. But now I can. I can go and look and say, hey, this person was treated with this drug. Is that, is that important? I can test that out. It's amazing. And just having the access to these data, like I said before, this is the largest data set available for this sort of data, and it's owned by all of us, right? It's our community participants. We are all together researchers and participants in this. And so, next slide. So, I want to talk a little bit about if there are any researchers online or researchers here, about how hard it is or easy to get and get started. So, and let me just say, it's not hard at all. So I just want to kind of provide a little bit of background. So, next slide. So I want to kind of talk about the tiers of data access, right? So there's 3 tiers. The first one is the public tier, which is available to everyone. You can go on right now and look at the public tier and essentially aggregate the double data from the data browser and look and see, hey, what's there? Is there something from the aggregated double? You can then, if you're like, okay, I want to look at actually more individual data, survey that. You can join the registered tier. And that's available to approved researchers through the research workbench. And that provides electronic health record, access to the electronic health records, surveys, physical measurements, taken enrollment. It's a lot of information. Well, if that's not, if you need to control data, right? That's available to approved researchers through the research workbench. And that provides access to genomics data, demographics data, for the electronic health records, and also like dates. So the diverse group, let me tell you kind of our experience with this. So we have this huge data set of, well, 500 individuals, not that huge, pretty small compared to this, but there's a lot of variance in there that we don't know. And so we're like, hey, where can we look? We spent 4 years trying to get access to a European data set that we look at. And we still haven't got it. We're still waiting on it. We're still waiting on this in order to be able to say, hey, wait a second, there's variance. Well, it took us less than a week to get signed up, go through the training, get our research program, our research question approved for a research workbench. Go through on the research hub, they have a data set builder. We could say, hey, we need genomics data. We need to be able to see if these variants are in this population. We were able to then import that in and say, okay, well, let's see how many patients there are or individuals there are with gastric cancer. They have that genomic data. We're able to then import that directly into our workspaces, which meant that, hey, now we're able to work on it. And they automatically started to generate a notebook. Now, these notebooks are essentially a computer on the cloud that allows us to, using our programming languages of R or Python, to be able to quickly access and query that data. Let me tell you, this is so accessible, right? So it cuts down the barrier of access from, okay, I need to be able to spend the next 5 years collecting the data. I'm going to have to get grants to fund, to be able to sequence it. I'm going to have to get $30,000 essentially for computer access, right? To be able to program it or to be able to handle the data, store the data that I want. None of that, right? I go on, we went on their server, they provide the infrastructure, they provide the resources to be able to analyze it. Now, was there learning, a little bit of a learning curve? There was. I was used to working within my computer cluster, my computer system, right? And so I think it took us a few days to figure it out, like I'm just like, oh, okay. But you know what? What we found, and it's kind of cut off here, was that there is so much documentation for researchers, so many examples. The user support hub, right, provides examples, documentation, videos, right? Every Friday, there's an open office hours where we could actually join and say, hey, we've got a question, or they provide, like, hey, this is how to research this. And so it really provides an easy way to get up and running on that. And that really democratizes the research, that it's not just left up to the researchers who have access to these $100,000 computers, but no, with a $200 laptop and a web browser, we can actually ask them, go in and say, hey, we've got a question. Let's find the answer to that. So next. So our research kind of moving forward, right? So we want to validate the previous findings from our smaller study, right? So we want to look in, we're looking in the data set. So when we looked a couple of months ago, we had 100 individuals that we were able to look, but you know what? We have to go back, right? Because April, they just released 250,000 more genomes. So, hey, this is a data set that we can continue to mine and come back to and see and help to do it for not just this study, but all the other previous smaller studies that we've done in breast cancer, colorectal cancer, all these things. So this really does allow us to identify and leverage those older studies that we found, or even the newer studies, right? To identify genetic risk factors for breast cancer, stomach cancer, and all the different kinds of cancer through analyzing all of this data. One of the things our lab is also interested in is helping to develop essentially models that doctors could essentially use to be able to identify a person's overall risk of cancer, right? So Caitlin mentioned before that a lot of these individuals come in with gastric cancer at stages, late stages. And the prognosis is bad for those. Well, what if we can identify, those individuals? The clinic, the doctor could say, hey, you have a family history. Well, let's do a simple, test and find out, oh, okay, if you're at a higher risk, we'll monitor you a little bit more closely so that instead of being caught at stage 4, you're caught at an early stage and be able to have that treatment available. And then also developing, that allows us to also start to understand the genetics, right? If we understand the genetics of it, then we can start to develop personalized cancer treatments, screening programs that really help address that health disparity. And everybody benefits, not just, we saw before, that those 4,000 are people that were like those 4,000, but everyone in the community can benefit from these discoveries. So, next slide. So yeah, just to end up, I just want to say that research is not like, it is a collaboration. It is a community collaboration and work that we all work together. And so I'm super excited. Caitlin and I are super excited to be here with all of us, and excited just for all of us moving forward as researchers. I wanted to say thank you to the people in our lab too. It's not just us. There's a lot of other people in the lab that are making these research possible. Lucha and our sister labs and our funding sources. But it's so wonderful to be here with you guys, participants today, and other researchers. And I encourage you, if you haven't already looked at all of us, please join. If you're a researcher, please join as a researcher, because there's so many questions we can answer so quickly with the resources that all of us made available. Thank you.
Activity is one I will remember for a really long time. That was so much fun. We'll save questions until after Hugo's talk, and then we'll have plenty of time for some bingo, which I'm really excited about. We also have some tables up for the people in person. And of course, the stall catchers activity to do some Alzheimer's research. But up next, we got Hugo. Hugo, the mic is yours.
Thank you.
All right, we'll go to the next slide.
Sure.
If you want to, wherever you want.
Sure.
And make sure you're kind of eating that mic. Okay.
Okay. Well, thanks everyone for being here. I appreciate you being here. And Fred, where do I go?
You can kind of stand up.
Over here.
Yeah.
All right.
Very good.
All right. So my talk is called "Understanding Genetic Research Results: A Patient's Journey." And
3 feet this way.
repeat.
3 feet this way.
Yeah, that
Oh,
way.
3 feet. This
Yeah,
way.
there.
There we go.
Yay.
All right.
Now you're on screen.
Okay. It's okay. I'll be here. The folks on Zoom can hear me. So this is my journey as a participant in the All of Us and as a participant in research. If you just advance to the next slide, please. So I've got a few caveats here for you. I am a participant and in research. I'm also a patient, a person living with a chronic heart condition. And I'm not a scientist or I'm not an expert. So this is really my experience as a patient, not the point of view of a scientist. So I hope I get the story right. All right, next slide, please. So what is my role and why am I here? So as I said, I'm a patient. I'm a person living with a genetic heart condition called hypertrophic cardiomyopathy, HCM. And an implantable cardioverter defibrillator. And the ICD is a device that's like a pacemaker. Pacemaker treats bradycardia, slow heart rhythms. The ICD treats tachycardia, fast heart rhythms. And so I have one of those devices, and it delivers a shock to the heart in case the device detects a malignant arrhythmia or an arrhythmia that could kill me. I'm also a participant ambassador for the All of Us research program. I'm really proud to be a part of that program. I'm a part of the Nutrition for Precision Health study, which is the first ancillary study to run on top of the All of Us platform. It's a very exciting study that's looking at this idea, this notion that nutrition is not one size fits all. It's also right here out of UC Davis. Really, really awesome study. And I'm part of PCORI, a Patient-Centered Outcomes Research Institute study called THRIVE, which is a pragmatic comparative effectiveness randomized control trial, which is a mouthful to say that's basically comparing 2 treatments for people going through having anesthesia. And so it's looking at intravenous anesthesia versus inhaled anesthesia from a patient's perspective. And there are a lot of folks like me, patients, much in the same way as with the All of Us and the Nutrition for Precision Health, folks like me who are patients, who are part of this study, who partner with researchers in the work. Next slide, please. So what is hypertrophic cardiomyopathy? By the time we're done here, you'll be experts in diagnosing HCM on people. I don't know, maybe not, but it's, so what is HCM? So hypertrophic cardiomyopathy is the most common type of inherited cardiomyopathy. It causes the walls of the heart to become abnormally thick, and it can put a person at risk for a sudden cardiac arrest. If you look at on the heart on the left there, it's a normal heart, but you can see that on the heart on the right, you can see the septal wall, the wall that separates the left side of the heart from the right side of the heart is abnormally thick. And so that is a hallmark of hypertrophic cardiomyopathy. Hypertrophy meaning it's thick and it's too big. HCM is the most common cause of sudden cardiac arrest in people who are younger than 35 years of age. A lot of times people go have no idea that they even have the HCM until there is some bad thing that happens to them. They pass out for no reason, they have a ventricular arrhythmia or suffer a cardiac arrest, which is what happened to me at age 37. Not an arrest, but an unexplained syncope and various other complications. All right, so move on to next slide, please. A little bit more about HCM. HCM is the most common, as I said, the most common type of genetic heart condition. The exact prevalence of HCM in the general population is not yet known. The number that folks kind of throw around is 1 in 500. But there's been, this is based on old echocardiogram data, and nowadays we're finding that it's probably a lot more prevalent. There's some studies that even said that are finding that the prevalence of HCM in the population, in the general population, might be even 1 in 200. So not a rare disease at all. Now, if you take into account the population of the US, the number of estimated people with hypertrophic cardiomyopathy may be about 700,000, but only about 100,000 people have been identified as having HCM. So there are a lot of us going around there with the disease not knowing, or perhaps with not the correct diagnosis. The disease, as it progresses, it can cause a variety of other health problems like atrial fibrillation, heart failure, systolic heart failure, diastolic heart failure, and like I said, a variety of different kinds of arrhythmias, atrial arrhythmias, ventricular arrhythmias, and sudden death. Now, HCM is an autosomal dominant condition, and I actually had to look that up because I didn't know that until I prepared the slides. And so I did look it up, and it means that there is a 50% chance of inheriting the condition from your parents if one of them is a carrier of a gene mutation that causes HCM, which also means that any of us would pass the disease. There's 50% chance of us passing the disease to our offspring, to our children, if we have a mutation, right? So let's go to slide 6, please. Okay, so HCM is primarily the reason why I participate in research, and this is why I got really involved. So this a crisis, I realized that I had a problem, and I engaged. But there are many reasons why people participate in research, and here are some of the reasons that people participate. Some folks may help, may have a sort of an altruistic desire to help advance scientific knowledge or contribute to medical science and improve the health of future generations. Others may have a personal interest in the condition or the disease that is being studied, which is the case with me with hypertrophic cardiomyopathy. Some other people may find it reassuring to have close monitoring by medical professionals in case of clinical trials and studies that require clinic visits. I have participated in clinical trials for drugs for hypertrophic cardiomyopathy because it wanted access to the doctors, and it was from a center that was not covered by my insurance, and And I wanted access to the echocardiograms that the study protocol required, and all of these things. So it is a possible reason why people participate in clinical trials. Others may seek access to to knowledge and experts. I find that my participation in the All of Us and in all the other studies that I'm a part of, I have the opportunity to be side by side with experts in the field of nutrition, with experts in the field of genomics, with experts in the field of cardiology, and being the dimmest bulb in the room, I feel like I can learn something from them, and that makes me a better patient, and it empowers me to have better conversations with my clinicians, and I learn a thing or two that I find useful to me. And some others may want the opportunity to receive life-saving treatments that may not yet be available to the general public. If you think about, for example, certain types of cancer, a person may have a scary diagnosis of stage 4 cancer and exhaust the possibility of treatment, and they may want to sign up for a clinical trial to have some hope to get better and feel better and have a medication that works for them that may not yet be approved for the general population. And finally, there's financial compensation, which is a valid reason, not really a reason I don't think a lot of people join studies because of that, usually a nominal fee, not really something to go laughing all the way to the bank about, but it's something that it makes you, it is a valid reason. I'm not here to judge anybody. All right, so next slide, please. So why join the All of Us? And so one of the benefits of joining the All of Us is to get your DNA information back to you for free if you want it. And this is kind of unique. A lot of studies usually don't return a lot of results to participants, but the All of Us does. And I find that useful. And so some of the information that the All of Us will return to participants is your genetic ancestry. So you may learn a little bit about what your, I don't know, where your family might come from in past hundreds of years ago. You may learn a little bit about your own genetic traits. And so why, things like whether or not you like cilantro, or the type of earwax that you have, whether you're lactose intolerant. You may not need an expensive research study to tell you those things. You may know that you're lactose intolerant or not. But it is interesting. It's kind of fun and interesting information to get back. But what I really find useful are the last two bullets there. Whether you may have an increased risk of developing a serious health condition, such as certain types of cancer and heart disease. And the last bullet there, another information that the program will return to you is how your body might react to certain medicines. And I think the term was mentioned earlier today, the pharmacogenomics or pharmacogenetics. I hope I'm saying this correctly. Those are very interesting and insightful things that you can learn about yourself by participating in the All of Us. Next slide, please. So for me, All of Us offered a chance to learn why I have HCM. It was there for me to take it. I never had genetic testing before, and when I was first diagnosed with HCM some like 15 years ago or so, I was offered genetic testing, but it was not covered by my insurance, and it was really expensive, thousands of dollars. And nowadays the price has come down, but it's still, there is, price of hundreds of dollars to get genetic testing. And so for me, at the time, it cost a lot of money, and it wasn't covered by insurance, so it didn't make sense for me to do it. I was also kind of in a different place in my journey with heart disease, and I didn't think it was important, so I declined. But now with the All of Us, I said, why not? I'll sign up. And so I did. I'm going to tell you a little bit about, next slide, please. Yes, thank you. A little bit about HCM and how HCM is a genetic condition that is caused by gene mutations in the cardiac sarcomere. We're going to get a little technical here. I had to research this stuff, I tell you, because I had heard the term, but I didn't really know exactly what this meant. So what is the cardiac sarcomere? The cardiac sarcomere is a part of the cardiac muscle that is responsible for the heart's contraction and the heart's ability to relax. It is a functional unit of the muscle cell that's driving the contraction of the muscle. A sarcomere gene mutation will cause the thickening of the heart, and it will cause HCM and may cause other cardiomyopathies. Now, to date, scientists have identified about 1,500 mutations affecting more than about 30 genes that are associated with HCM. So this means that a gene mutation, an abnormal gene, will that has to a sarcomeric gene that codes that helps with the that has to do with the cardiac sarcomere will have an impact on how the heart contracts and how the heart relaxes. Typically, HCM hearts contract really well, but have trouble relaxing. All right, and next slide, please. I want to point this out. I just want to put this in the back of your minds there, because I'm going to come back to this slide, not the exact same slide, but this idea about disease as we talk about the results and the pharmacogenetics results that I received from the program. But I want to say a couple of things about drugs used to treat HCM. Historically, HCM patients have had limited medical therapy options with drugs that were not developed specifically for HCM. So patients were prescribed beta blockers, and the drugs that I have listed there, like atenolol, metoprolol, propanolol, sotalol, and all these drugs that end with all are beta blockers. Or patients would be treated with calcium channel blockers, like I have a couple of examples there, verapamil and diltiazem. Or they would be given heart rhythm drugs if the person has heart rhythm disturbances, like amiodarone, disopyramide. Or if they had atrial fibrillation, for example, if they were having an issue with risk for stroke, they would be put on blood thinners like warfarin, the bigatron, like Pradaxa and Eliquis, and a variety of other drugs like that. But we're now starting to see a whole new class of drugs that have been developed specifically for hypertrophic. They are being developed specifically for hypertrophic cardiomyopathy, and that's what you see there on the right side of the screen. There's a new drug called mavacamten that was just approved by the FDA about 12 months ago, April 2022, I think, and this drug targets the cardiac sarcomere, and that is why I mentioned the cardiac sarcomere. So it's an entirely different class of drug specifically designed for HCM. So and it's working really well. It makes a lot of the symptoms go away. People are doing really well on this drug. We don't know what the long-term outcome will be, but hopefully it's at least certainly something that gives great hope for people who have failed the other therapies. And so, I think it's kind of this whole thing about precision medicine is opening up this entire new era of precision pharmacological treatment for many diseases, including HCM. So keep the thought about the mavacamten and that drug in the back pocket, because we'll come back to it in a few slides ahead. Next slide, please. So all of us looks at a list of 59. So regarding your actionable, medically actionable results, all of us looks at a list of 59 genes that are considered medically actionable, and this list is called the ACMG 59. And ACMG stands for American College of Medical Genetics. And these are a list of 59 genes that are known to be associated with conditions that are inherited, like some types of cancer and heart disease, all these cardiomyopathies. So there are, in this list of 59 genes, there are about 15 genes that have to do with cardiomyopathy, like hypertrophic cardiomyopathy, dilated cardiomyopathy. I put the list there so that you have a sense of, it's not the center of the picture in the center of the slide. It's not really to be read, but that's the entire list of 59 genes. On the left there, you see hypertrophic cardiomyopathy. There are a variety of genes there for that dilated cardiomyopathy and arrhythmogenic right ventricular dysplasia or cardiomyopathy, I guess the name now for the diseases. So these are inherited types of cardiomyopathies that some of them share some of the same genes. And so there are 15 genes that have to do with these cardiomyopathies. Next slide, please. However, when you talk about HCM, there are, among patients that have the diagnosis of HCM, people like me, nowadays we know that only about 33% will have a pathogenic, will find a pathogenic variant in their genetics, I suppose. And so as the science has advanced, this percentage has been getting smaller and smaller because we're finding that there are so many genes that cause HCM and these other cardiomyopathies that we used to think that for each patient with a diagnosis of HCM, we would find a pathogenic variant in 50% of them. That number went down 42%, now it's 33%. So as our knowledge about genetics expands and program studies like the All of Us and many others, as we expand our knowledge about genetics, we are finding new variants. And this is good news and bad news. So for me, I kind of, sort of reduces my chance of having a genetic variant in one of those 15 genes in the list of ACMG 59 that the All of Us looks at. But I was hoping that I knew it was a bit of a long shot, but I hoped that the All of Us would find the genetic mutation that caused my HCM. Next slide, please. So let's take a look at my results. And we're gonna go quickly through these slides. And next slide, please. So this is a screenshot of the participant portal. I mean, all of it, and I guess 99% of this is amazing. By the way, I'm really proud of you guys for having joined. No, you guys are incredible. It's a large group here of participants in the All of Us. So you are familiar with the participant portal. So once you log into the participant portal, next slide, please. You'll see the screen, and there's a little button there where the arrow is called My Data. If you click on that, next slide, you get to this page where you see the button there up on top. This is where your data is, and the button there up on top says DNA results. And if you click on the DNA results, next please, you get to this page, and you see that on this page you have all of your DNA related results will be on this page. What I want to talk about, I want to focus on the two top results there, the hereditary disease risk and the medicine and your DNA, right below there, the little icon below that, medicine and your DNA. That's what I'm not going to focus on anything else. And you can see there's a button there to do results, and there's a button there to schedule your appointment, and that's to schedule an appointment with a genetic counselor, which by the way, I find that alone is incredibly valuable because that's an expensive consultation, but you have free access to that as a participant in the All of Us when you get your results. The next slide, please. So I was surprised to find that horrible, right? And I was so hopeful that I was going to find a reason why I have HCM. All right, this is very disappointing. I was very disappointed, and I know it was a long shot, but still, one can hope. And I thought, and it wouldn't, and this wouldn't change my diagnosis. I still have HCM. It only means that the All of Us didn't find in those genes for known for causing hypertrophic cardiomyopathy within the ACMG59. No known mutations found. So so this was a little disappointing, and there might be some reasons why that is true, that this happened. I spoke with a genetic counselor, and she gave me 3 possible reasons. There may be more reasons, but let's take a look at some of the reasons why these results were negative. Next slide, please. Number 1, maybe the test just simply did not detect an existing mutation in one of the genes that are known to cause HCM. So what I was told by the genetic counselor is that this is a research test, and because it is a research test, there are limitations, and a clinical test may be more specific and may be more comprehensive. And so it is possible that if we redo the test or have a clinical grade test done, I may get a positive hit in one of those genes. So this is one possibility that why I got a negative result. A second possibility is that that I have a mutation in one of the many other genes that are associated with HCM that are not included in the ACMG59 panel of genes. So if you remember, you may remember I said something like there are, well, I didn't say something, I said exactly this. There are 30 known genes, roughly about 30 known genes, 1,500 mutations that are known to cause hypertrophic cardiomyopathy. And so it is the ACMG list of 59 genes includes about 15 the genes that have to do with cardiomyopathies. I think 9 or 10 perhaps that have to do with HCM specifically. So it is possible that I have a mutation that was just not looked at, and that's why we didn't find it. And the third possibility is that I may have a mutation in the gene that is not yet known to be associated with HCM. And these results, even though they're negative and not exactly what I wanted to see, it gives me some hope. It gives me hope that my genome is one in those 250,000 genomes that have been sequenced. Hopefully soon will be 1 in a million. And that it's there, and for researchers and geneticists like you guys to one day come back and look at it and discover new new variants that cause the disease, that cause hypertrophic cardiomyopathy. All right, so next slide, please. We're wrapping up here. But all of us, so this is where I think the surprise comes, for me at least it was. All of us also looks at, I think, 7 genes that have to do with the way your body processes or metabolizes medication. And I didn't really have any, I didn't know, I wasn't expecting anything in particular with that. I wasn't even thinking about it that much. I was really focused on the genes that identifying the gene that caused my HCM. I wasn't thinking about my drug interaction or how the genes metabolize my drugs. So when I got these results, if you skip to, no, before you go, let me just mention this. This is important. And we talked about this is pharmacogenetics. So these genes that have to do with the way you metabolize medications do different things. So they either move medicines to the right part of the body, or they break down medicines and clear them from the body. So there are different functions that these genes could do. Or change medicines into a form that can be absorbed or they can make them work better in the body. So there are different functions. So next slide, please. Let's take a look at my results. So to my surprise, all of us flagged a couple of different genes in which I am a poor, an intermediate metabolizer, and one of the genes has a poor function. Next slide, please. So we're going to go into these a little bit. So if you look on the left there, these are the 7 genes. I'm not going to show you all the others because they're normal. So nothing particularly interesting about them. But these 2 genes, CYP2C19 and SLCO1B1, One of them, the one in the bottom there, I have a poor function. The one on top, I'm in an intermediate metabolizer. I'll pick, let's start with the one in the bottom, SLCO, or I'm not sure if it's even a 0. I think it's a no. 1B1. That's how much about genetics I know. So this is a gene that helps facilitate the uptake of statins. And I'm sure many of us here have heard the term statins. It's a medication that prevents heart attacks, reduce your risk for heart attack. And so I am a poor metabolizer of that gene. So this means people with changes in this gene will have a harder time removing these medicines from their body. This means that those medicines may build up in the body and cause muscle pain. So I know something. Now I'm not taking any statins, but now I know something that if I ever am prescribed a statin, I'll be having a conversation with my doctor about this, because I know that there are medications, statin medications that are designed for people who are poor metabolizers of this gene. So this is a useful information to have. Instead of just, because you would never know if you're taking a statin, you may not even know if it's working, since it's a prophylactic drug. It's there just to prevent something bad from happening. You may think, oh, it's working. It's probably working. And there's no way of knowing if it's really working unless you get genetic testing. So I thought that was kind of interesting. Not directly related to my care, but useful. Now, the other one is directly related to my care, and I thought that was interesting. So that's the CYP2C19. And that's the medication. You may remember the medication I mentioned earlier, mavacamten, one of the new drugs that works directly, that targets the cardiac sarcomere. That drug, and it's a new drug, and in fact, my doctor and I had conversations about it. She says, you may consider, and it's a very expensive drug, and it's a new drug, and so she says, well, maybe in the future we're talking about putting you on that. And turns out this drug mavacamten is primarily metabolized by this gene, CYP2C19. And since I'm an intermediate metabolizer, there is a possibility it may not work well for me. So this opened up the possibility, the opportunity for me to have a conversation with my doctor about this. I don't quite know the answer to that. I am not able to make a decision on my own, but at least it put me in a place where I can have a conversation with my doctor about this, and together we can make a decision. So I thought that was surprising and sort of unexpected for me. So I have one last slide for you, a conclusion. So these are, a few thoughts about participating in research. In my view, participating in research can help patients with chronic conditions stay informed and become better experts in their own health. I think it's done that for me. I'm a better patient because I participate in research and I'm constantly learning. Also, by actively engaging in self-education, patients can learn invaluable knowledge that allows them to better manage their condition. I think that's also sort of something that's happened to me. And finally, I think participating in research can be a very rewarding experience that offers each one of us a sense of purpose. And I feel like I'm preaching to the choir because all of you here are participants in the All of Us. So thank you so much for your attention and your time.
Fantastic. So we have our panel set up here. We're going to do a few questions before we get to bingo, which is very fun. So we'll leave that thank you up behind you while we talk. So I think I'll take one question from online and then a few from in person, and then we want to leave enough time for bingo and for our people in person to get some more food and look at our tables, because the library does close at 6 and they will kick us out. So we are on a deadline here. Okay, so question from the internet. The challenge is associating the genotypes with the phenotypes. How will these phenotypes be recorded over lifetimes in a standardized format? Who wants to take that? All right.
So the main way that we get phenotypes, that is what happens to somebody, what diseases they get, what conditions they show up with, what symptoms they complain of, is from the medical record data. And you can imagine like even when you go to your My Chart or your version of the medical record, it's very complicated. And so what we figured out how to do with the medical record data is we figured out how to take any medical record and put it in the same format so that all the data can be found in the same locations throughout. And computer tools are helping us do this better and better. So we can even take a doctor's note, which is, you know, written note, or sometimes they dictate it into the computer, and we can abstract from that features which are like the symptom that you complained about. And so then all that data goes into this big database, and those phenotypic changes are buried in there. It may be combinations of things. We might say a phenotype is somebody who has asthma and they complain, but they don't come in saying they have asthma. They come in saying they're having trouble breathing and they're wheezing. But then we also see what the doctor prescribed. The doctor may have prescribed albuterol or an inhaled a steroid drug. And so we put these pieces together and we say, aha, that's a person with an asthma phenotype. And then we can look at their genetics and we can look at their outcomes. Their genetics, we may learn, have special features that predict in the future that that's somebody who needs more aggressive treatment for their asthma. Or alternatively, somebody who just has a little bit of asthma and it's probably going to go away. So this is what we're trying to do a much more personalized job of treating individuals based on all those complexities of data.
Fantastic. All right, I saw you had your hand up, so we'll go to you. There you go.
Thank you. On one of the slides that you showed, you had the SLC01B1 as one of the results. I can't make heads or tails of what that means, even though I'm reading the description down below, because I'm comparing mine to yours, and my number was higher or lower on both of them to yours, like opposite, and yet mine still says something similar to yours, which made like no sense. So how do we figure, is it something that we bring to our primary care physician, or is it something that you guys can answer today?
All right, who wants to take it? All right,
I can start with this.
perfect.
I
Teamwork.
I can tell you, thank you for that question. I felt the same way. I was very overwhelmed and I had no idea what to make of it. So what was helpful to me was the call with a genetic counselor. So I did schedule an appointment. It was very, very easy to schedule, and I did schedule an appointment. And I spoke with it. I spoke with him twice, and you can schedule as many times as you want. And so so I spoke with the genetic counselor because I did not understand exactly what that meant, and I made a list of questions for her, and she explained and sent me a report. In fact, they sent me a written report afterwards and explained from our notes from our conversation. There were a report that was written specifically for me based on what we discussed, and sent me links to sites to learn more about it. So I think without the benefit of the genetic counselor, I would be very, very lost. I still don't think I understand a whole lot of things, but I understand a little more than I did before going through the process. So I feel like I've learned a little bit.
Yeah.
Once you do that, in all likelihood, you will be in a position to teach your primary care doctor more about the subject. As you, I'm sure are aware, like in the old days of medical school, we were supposed to learn everything. And the doctor was supposed to know everything. Those days are long gone, especially with all of these genes. And most doctors, like you, haven't heard of SLC 107 or any of these genes, or if they've heard of them, they certainly don't remember what that gene means and what your variant of that gene means. But we're trying to give them the tools to be able to look it up. And that's how we teach medical students today. We don't point a fire hose of data at them. We try and give them the tools to find answers when they need to. And it's been a real shift in how we teach medical students. But you, as an active participant in all of us, with your own data and an understanding of what that means, can be part of that education process.
Fantastic. I saw another hand right here.
For Hugo, the device you have in right now, what would be the difference with them if they put a pacemaker in instead of that?
Thank you for that question. I think I can answer that. So the pacemaker, the way I think of it is the difference between a pacemaker and an implantable portable defibrillator, or ICD, is that the pacemaker is there to treat slow heart rhythms, bradycardia. So the pacemaker is only capable of pacing the heart up. An ICD is able to, in addition to, all ICDs are pacemakers as well, by the way. But the ICD, in addition to being able to pace, it is also able to detect a ventricular arrhythmia, an arrhythmia in the lower chambers of the heart that can put your life at risk and deliver a shock that stuns the heart, and hopefully the natural pacemaker of the heart picks it up. And if it doesn't, the ICD paces your heart and saves your life. That's worked for me 4 times, so I wouldn't be here without the ICD.
That's what I have right now.
Fantastic. You want to go next? All right, and I think we'll take one more question from online and one more question in person, so we'll get you. Because we want to make sure you have time to chat, to eat. We have bingo still, so that's important. We have to play the bingo. We printed all these cards. So, question from online: does my DNA change if I develop a disease in my lifetime? Yeah. It's National DNA Day, so we have to.
I can. So yeah, so that your DNA is very dynamic, and your cells, anytime they replicate, there can be an error. So that's typically what we think of when we think of cancer. So if we're thinking of cancer, cancer is a very rapid change, and the big changes in the DNA, breaks, mutations, and leads to uncontrolled growth. But in typical cells, the amount of changes is very minimal. Although, I say that, the majority of mutations or variants that we inherit from our mom and dad. But even from them, I have a few a few variants that neither my mom had nor my dad had, just because of the fact that whenever DNA is copied or a cell replicates, or then that there's a chance for an error. And our cells try to fix all the errors, but oftentimes those can't be fixed. And so different times as well, like if we go out and if I get sunburn, right? That sunburn is actually the damage to my cells, and UV rays are mutating my DNA. And so my body tries to repair that, but as I get old, I have a family history of skin cancer. And so, I'm confident that my brothers, my dad, everyone, grandpa, I'm going to have skin cancer at some point because of that, those DNA changes aggregate over time. And I haven't been as careful as I should have. So, yeah, so it does a little bit, but.
Yeah, so broadly, we categorize DNA and your sequence and your genome into 2 categories. So one category is your inherited genome, and that's also sometimes called germline DNA sequence. And then the other, every cell in your body carries a complete copy. But as Paul just said, sometimes those copies get altered a little bit in the, on the copier. You take a Xerox copier, the old copiers, and you put a page on there, and then you put it back on again. And eventually, it doesn't look anything like it started. So the same thing is happening in all our cells. We call that somatic, meaning in our body DNA sequence, or somatic genomics, which is very important in cancer, but can be important in other diseases too. So it's a really good question. In the All of Us study, we're trying to get your inherited or germline genome in the record, and that's the primary interest. We're not, for example, taking a sample of anybody in the All of Us study who winds up with cancer and sequencing that. But that would be a good study to add on later.
That's pretty cool. Awesome. We got a question in the back, and then we're gonna get to our all of us bingo.
Thank you. I understand how you guys figure out what, so many people have a condition, so many people have that gene, obviously it's kind of relates to that. Is there no physical way you can tell with that mutation like you are that there is a gene involved in it? I guess not, because you said that you don't know what it is.
So you know, yeah. That's a great question. And that kind, yeah, so there is a way, and a little bit of a way anyways. In some cases, we can take that gene and mutate it just like we saw and see if that affects the function of that gene, measure the function of that gene in the lab. That, however, is like, you know, ends up being even longer, right? Years and years to be able to quantify those changes and to then, you know, make sure that, you know, what we see in the lab is actually happening in, you know, our bodies and that. So the faster way that we can, you know, just show that is associated essentially is to look in the population and to see, yeah, that it's more, you know, represented in this population versus this, then it must be affecting these individuals. And so that's the initial kind of quicker way to look. But yeah, ultimately, yeah, to understand like what it's doing, and that is so sometimes go back into the lab and make a copy of that and let's see if we can do it. So for some genes, that's a lot easier. For some genes, that's a little bit harder because the effects may be small and hard to measure over time. Whereas, yeah, they have an effect over a long time versus just something. So yeah, it's a great question.
Impressive. I lied. There's another really good question online, so we're gonna take it. So someone said 3 in our family have been sampled for all of us at UCSF over a year ago and did single samples for blood, urine, etc. But Professor Snyder at Stanford and others are looking at using wearables, real-time data, steps, pulse, PO2, etc. When might all of us give the option to track all of this data to give leading indicators of pending illness? All right.
So all of us does connect to your wearables, although we only have Fitbit so far. I think we're just about to have Apple Watch. One of the projects that we're about to start that Hugo and I have been working on for the last year is a nutrition study. So this is an additional study where we're going to measure many more things, including physical measures. So, you know, how easy or hard it is for you to stand up from a squatting position, and all kinds of things like that. And also to track where you go, track what you eat over at least a 2-week period. And then we're going to challenge you with this milkshake that we concocted so that we can see what happens in your blood. Are you a fast metabolizer of fats and lipids, or are you slow? Are you a fast metabolizer of fructose, or are you slow? And so forth. And so there's all kinds of opportunity in all of us to do the kinds of things that I guess Dr. Snyder's doing at Stanford. He and others at Stanford will also be able to tap into this database once we get all the data assembled from the nutrition study. So there's lots of good stuff going on, lots of ways to combine big data sets, and this is all going to benefit our health.
All right.
Play
So,
some bingo.
yeah, we'll play some bingo, and I want the panelists to play bingo too. All right. And then the conversation doesn't have to end here. We're stopping questions now, but if you follow All of Us California on social media, All of Us CA, you'll be able to join events like this one, stay up to date with the latest and greatest in All of Us. I know the Sacramento Public Library has a lot of great health-related events coming through, so follow them, follow us at SciStarter. But for today, if you're online, go to the next slide. We're gonna play some bingo. So for those of you, you can download this document if you're online and play with us. We're gonna play until we have someone who gets a bingo. So that's when you have 5 squares in a row, horizontally, vertically, or diagonally. We have some cool prizes. For those you can't win the prizes if you're online, sorry guys. But if you're in person in the back, Elizabeth is gonna be the person distributing your prizes. Oh, it's a water bottle. Wow, I really want that. I wish I could play. Okay, so our first question, or not question, prompt. So I want you all to clasp your hands without thinking about it. So clasp them. All right, everybody's clasping. So color the square. I clasp my right hand over my left if this describes you. Who put their right hand over their left? All right, you did color that square. Color it on the bingo board. If you don't have pins, we have pins on the side. Okay, I see people coloring. Great, great, great. All right, next prompt. Color the square marked I like the taste of cilantro if this describes you. For those who just came in, we're playing bingo. So you're welcome to play with us. We're going to talk about citizen science in just a second. We just want to chill until then. Yeah, feel free to take a seat. We're so glad you're here. And here, I'll put the bingo up here in case you want to grab it. Okay, so our next one is color the square marked my second toe is shorter than my first, if this describes you. For those of you wearing closed-toed shoes, you can take them off if you want, or not. You can just use your memory. Next we have color the square marked I have allergies, if you have this trait.
Very broad.
Color the square marked I don't have allergies if you have this trait. So if you don't have allergies, you can color that square. And remember, y'all have a free space. That's always a big part of my strategy for bingo. Okay. Find the two squares describing earlobes and color the one that describes you. So the earlobe squares, color the earlobe square that describes you. Do we have any bingos yet? We've gone through more than 5 prompts, so we could feasibly have a bingo. So find the 2 earlobe ones and color the one that describes you. All right, we've done earlobes. Now let's do hairlines. So find the 2 squares describing hairline and color the one that applies to you. Do we have any bingos? No bingos yet? Okay, we'll keep it going. All right, color the square marked I dislike the taste of cilantro if this describes you. Is there anyone here who thinks cilantro tastes like soap? Okay, I see a few people. You color those squares. All right, find the 2 squares describing hair texture, curly or straight. Color the one that applies to you. If you have curly hair, color the curly haired square. If you have straight hair, color the straight haired square. Color both. You can identify with both types of hair. All right, color the square marked my second toe is longer than my first if this describes you.
What?
Color the square marked my second toe is longer than my first if this describes you. Do we not have any bingos yet? Oh my goodness. All right, we'll keep it going. Find the 2 squares describing freckles and color the one that applies to you. Freckles. Color the freckle square that applies to you. I'll pause here. I know people have been coloring a lot of things. And you know, when you get bingo, you can shout bingo. We're ready to have a winner. All right. Now, once again, I'm going to ask you all, clasp your hands without thinking about it. Go ahead and clasp them. All right. I want to see some hand clasps in here. Clasp your hands without thinking about it. Okay, perfect. Color the square marked I clasp my left hand over my right. I clasp my left hand over my right, if this applies to you. And the picture does help. We got a helpful prompt from Dr. Borowsky at the front. Okay, find the 2 squares describing tongue rolling ability. Tongue rolling ability. I don't have this, otherwise I would demo it for you. Color the square that applies to you. Look at the tongue rolling ability squares and color the ones that apply to you. All right, find the 2 squares describing dimples and color the one that applies to you. Dimples. Color the dimple square that applies to you.
I got it.
We got a bingo! Bingo! We have a winner of the water bottle. People on the internet, I know you are jealous. I know you are very jealous, but that's okay. We're just happy you're here. All right, so some things about this activity, right? These were all observable traits for the most part, like eye color, height, hair texture, etc. Some of you share traits in common, but the combination of your traits is unique. And the connections to all of us, right? Some inherited traits involve our health. For example, the risk of developing some diseases is a trait. Our traits are determined in part by our DNA, and the All of Us research program collects DNA samples to help researchers learn more. Since traits are inherited, participating in the All of Us research program could support research that helps future generations. So for those of you who participate in All of Us, which is pretty much everybody in person here, and quite a few people who are watching online, you can feel good about it because you know you're helping future generations as well. We're doing something for our community. If you haven't participated, you can go to joinallofus.org and you can get started. So we'll go to the next slide. So this is another document. We really encourage y'all to do this at home. If you want to download this document, it's more of a handout that, if we go to the next slide, relates to a video that you can also watch at home. We're running a little short on time, so we won't watch it in here. We'll go to the next slide. So if you all did All of Us, you participated, and you're like, wow, I really like this science thing. This is really cool. And you're hungry for more. This is a separate citizen science project called Stall Catchers. I just wanted to share with you. It's separate from All of Us, but something at SciStarter that we support that I think you all might enjoy. So basically, there's 30% less blood flow in a brain with Alzheimer's. So we'll go to the next slide. And the reason why, this is a finding from Cornell University, stalls. Clog blood vessels in the brain, stall the blood flow, and that contributes to Alzheimer's disease. We'll go to the next slide. So a novel finding from these researchers is that restoring blood flow restores memory and reduces other symptoms of Alzheimer's in mice. So this is super noteworthy, right? Stalled blood vessels, the resulting loss of blood flow contributes to Alzheimer's, but restoring that blood flow reduces some symptoms in mice. So we'll go to the next slide. Keep going. So it takes 6 to 12 months for this team at Cornell to answer one research question in the lab. Next slide. But with Stall Catchers, which is a citizen science project that everybody in this room, everybody watching online, anywhere on Earth, it's a project you can do to help. You can help analyze these data for the researchers. You can help them understand and answer different questions about Alzheimer's disease by catching these stalled blood vessels. We'll go to the next slide. So there are over 50,000 catchers from around the world, which you all can join their numbers if you play. Next slide. There are 50 to 100 playing every day, classifying this data, analyzing this data to help these Alzheimer's researchers. Next slide. It speeds up the research by 5 times, so we're able to answer these important questions faster. We all want a cure for Alzheimer's. This project is helping us understand this disease. We'll go to the next slide. So now it only takes 1 to 2 months per research question with the Stall Catcher citizen scientists. Next slide. We'll skip this video actually. So, because I want you all to just play it yourself. So you just go online, you watch these videos, you say whether or not the blood vessels stalled or flowing. They teach you how to do it with a few training videos, and then you get right into classifying the data. I've seen middle schoolers do this. I've seen retirees do this. Anybody can do Stall Catchers and classify the data. And by doing that, you're part of the team analyzing it, and you might end up on a co-author on a publication. These are all publications, research findings related to Alzheimer's that this project has accomplished, and the Stall Catchers contributors are cited as co-authors, which I find really exciting because you all are partners in this research as citizen scientists. You're helping do the analysis, which is super cool. We'll go to the next slide. So the main goal of this project is to piece together a molecular pathway to understand Alzheimer's. I also think another cool thing about citizen science is you don't have to know every single detail about Alzheimer's disease to be a meaningful part of the analysis. I can't answer questions. I can't really describe this graphic for you, but I can play Stall Catchers. I can conduct the analysis. I can identify what blood vessels are stalled and which ones are flowing. And by doing that, I know that I'm contributing to this research understanding. I'm helping those researchers who are analyzing this data to understand the disease and hopefully find a cure. So next slide. So if you want to participate in Stall Catchers, you can go to SciStarter.org/NLM. I'll also be lurking in the corner for the last 10 minutes of this event if you want to demo it with me. But we'll go to the next slide. But in short, I just wanted to thank everybody for speaking today, for the All of Us research program, for the Network of the National Library of Medicine, sharing this information with you all. I learned a lot about DNA. I think you all did too. I learned a lot about how you can be hands-on with your health. With the All of Us research program, and how you can help people in future generations by participating. And also, I shared a little citizen science project with you all called Stall Catchers that you can do at home if you want even more of this science. So that's the close of today's event. For the people online, you can keep on commenting in the chat if you have questions. I'll be answering those, by text in the chat. Otherwise, for the people who are here in person, please grab some more food, check out the tables. If you haven't joined all of us yet, go to joinallofus.org/CA for the California one. We also have our Citizen Science Month link. These are all different tracking links. They all go to the same place. We'll go to the next slide. You can also potentially win a free book if you fill out our survey. It's links.asu.edu/CSN-participant-survey79. That's evaluating this event. I probably am not supposed to tell you this, but response rates have been really low, and we have 50 of these books. So if you fill out the survey, you'll probably win a book. It's the Field Guide to Citizen Science. My boss wrote it. I liked it. It's a good book. So feel free to fill out the survey if you probably want to win a copy of this book. Otherwise, thanks so much to our panel again, and we'll be here in person, and the online people, I'll be communicating in chat. Thank you so much. First, really quickly. Okay. Yeah. We'll just leave this up to the online people. Thank you so much for sharing.
Sure.
This is only tangentially related.
Oh, I know.